So I will speak about S.A.F.AN bioinformatics technology and how we use it to fight, to study a new solution for COVID-19. So who we are a small bioinformatics company having one of the most innovative in silico profiling technology, and to support initial drug discovery and repositioning project. And our mission and our ambition is to give a prediction of a protein compound binding so reliable that finally in vitro test will be just a very a check, but not really needed. So how it works, there is a small molecule or a peptide, and we get out with a target profiling with experimental reliability. And then we can check with the side effect database linked to targets or to disease databases linked to databases. And this goes to drug repositioning or to safety issue. Which are the target classes that we can predict? As you see, there are many target classes, most for small molecule than for peptides, but all the most important target classes are included as enzyme, membrane receptor, ion channel, transporter, transcription factor, adhesion, epigenetic, secretic, structural, and so on. So a short slide on the validation of this technology, why we say that is so precise, that is almost close to experiment, is we did a validation for small molecule on 21,000 small molecule, which means 66,000 of data. And the correlation between the prediction and the experimental data is 91%. For peptides, there are fewer data available. So we did it on 4,382 molecules. And the data are about 6,000, and the correlation is 87%. So now, as you know, for the COVID project, the starting of the process is when the virus enters the cell and it binds to AC2 receptor, and it has to be activated by proteases. So membrane protein proteases, and it has also recently found that the spike protein has a furin site, so also furin could be involved in the process. So on one side, we'll look for compounds using our technology, S.A.F.AN ISP, that could bind to the proteases. And then we will verify this binding and the detail of atomic interaction by structural bioinformatics. And on the other side, by structural bioinformatic techniques, we will use a protein-protein interaction protocol and to find the molecule that inhibit the interaction between the AC2 and the spike protein. And then by chemoinformatics, we will check which are the off targets of the molecule that we find. So what about the S.A.F.AN ISP that we use for COVID? We already screened 3 data sets. One is those of the clinical stage compounds found on Campbell, and there are about 6,000 compounds. And for that, we found 42 compounds that display a prediction, submicromolar prediction to the interesting proteases. Then we screen a database of natural compounds of 36,000 compounds, and from there we find 28 interesting compounds, and a database of food compounds which contain 26,000 compounds, and from there we found 51 compounds. So we didn't do it yet, but the next step will be to check with the furin and the protein, the serine membrane proteases, the docking process, and the molecular dynamic simulation to check the binding and to check the stability of the binding and the atomic details. So for what concern the protein-protein inhibition, the protein-protein interaction inhibition of AC2 to the spike protein, the complex of these two molecules has already been solved by protein crystallography, and the structure is available from the protein data bank. So it is possible to use this structure as template to find a compound that could bind to the AC2 receptor and inhibit the interaction with the spike protein. So which is the protocol that we used? We took the protein, the crystal structure of the complex, and by visual inspection, we select the interaction peptide, one of the two interaction peptide, and we made a template model. So then we took the database of the clinical stage compound, those for which we already had the S.A.F.AN ISP prediction. And with the program Yazara, we did an atom superimposition of all these compounds to the template. And we select those that had, for which the complex was with energy below 0, and they had a certain number of atoms matching, and they had a certain number of hydrogen bonds. So from that, we selected 248 compounds, and we refined, we did a further selection, and we refined 42 compounds with 500 picosecond on molecular dynamic simulation. And then from these, the analysis of these 42 compounds, we select 14 compounds, and with a longer simulation, which is not as long as people do now, but still is able to show if the compound still gets stick to the AC2 receptor, or it goes away during the simulation. So from UniProt, from the data available, it is known that there are some position in the AC2 receptor that are particularly important for binding the SARS-CoV spike glycoprotein. And some of them abolish the interaction, some of them strongly inhibit the interaction. So we checked what happened in the new SARS-CoV-2 complex. And we found that only for position 353, there is still a hydrogen bond. The other, there are several contacts, but no hydrogen bond. And so we decided to use this as a driving idea to select compound and interactions. So this is the analysis of the short simulation. So of the simulation used to refine the complexes. And we see that in some cases, the contacts with the starting, the amino acid that were originally in contact with the compounds are quite well conserved, but there are no hydrogen bond. And in other cases, they are not conserved at all. And in other cases, the hydrogen bond to lysine 353 is very well conserved. So this is how we started to look for the new steps. Then we also did a simulation of the mutants that are described to inhibit the mutant of lysine 253 that are described to inhibit the interaction with AC2 in the SARS-CoV complex. And we see here in this slide that with the exception of compound 199, in all the other cases, the contacts are much better conserved in the wild type than in the mutants. So indicating that the lysine seems to be really important for the binding. To analyze the long simulation, we thought that we need to have a positive and a negative control. So we went to look in literature, and we found that there are, of course, already some compound that inhibit protein-protein interaction, and in some cases the crystal structure exists. And so we choose the compound that inhibit the interaction between E3 ligase VHL and IF1A. And there is a positive control that has an IC50 of 3.8 micromolar, and which is the crystal structure, and the negative control, which has an IC50 of 270 micromolar, that is not present in the crystal structure, but we generate just mutating the structure. So these are the results of the protein-protein inhibition from the long simulation. And you see that in the positive control for 28 nanoseconds of simulation, the contacts are very, very well preserved, while in the negative control, they are almost lost. What we see also is that in some cases, in some compounds, the contacts with the hydrogen bond to lysine 253 is quite well preserved all along the simulation. And in some cases also, all the contacts are preserved more than in the negative control. So these compounds probably are interesting for further analysis and experimental evaluation. So what next? The experimental validation in collaboration with Humana Biosciences in our programs. And finally, thank you for your attention.